we'll start with some preview for the discussion this week. This should be your last exercise type of discussion that we've had. We've kind of built off of the joints. We had a couple discussions on joints and one on muscles, and now this will be your last muscle one, because after this week we'll be starting the nervous system, and that's our last system of the semester. So we'll be spending essentially a month on just the nervous system. And there's a lot in the nervous system, which is why we spend so much time on it. All right, so I guess kind of as a discussion preview, who could run a marathon tomorrow? Jack, you want to go for it? Matt? Have you run one before? Have you run one before?
Yeah, I've run some ultras before.
Oh, okay, so.
I know how to pace myself, so.
Okay, you could probably walk one. But essentially the question is, how do you get in shape and what does that mean for your muscles? Okay, other than what we talked about last week, which was actually building the muscles bigger, right? What else is going on in your muscle physiology and anatomy that allows you to run a marathon where you wouldn't be able to before? Because essentially, a lot of the things are the same, right? Your muscle's the same, the processes are the same, but how come you can run 26 miles after training for 6 months, but you can't just turn around and do that? Yeah?
Oxygen absorption.
Okay, so this is, again, that's a good part of it is your capacity for oxygen. But what I want you to do, of course, is expand on the things that you've learned in lecture, but go to the scientific literature and show me studies where they've found this out. And what does it mean? And how does your muscle change? Okay? All right, so that's the discussion. For a couple other things on there. So I did post a blog that somebody wrote about somebody who ran an ultra marathon or an Ironman marathon, not marathon, triathlon, without training at all. So he just decided one day, I've got an open Saturday, I'm going to do a ultra triathlon, which is like a 100-mile bike ride, a marathon, and then like a 2-mile swim or something. 4 miles, right. So I don't know if I could ever do that, but definitely not without any training. But the experience yeah, you did it. Yeah, it's kind of funny to read through and talk about and hear the whole experience. But essentially, he did this without being in shape to do it, and so there are some problems that can occur if you try that. But I would read. It's a good read. I would read through that and then do some research. This week, all right, so today the motor unit. What is a motor unit? It involves our nervous system, right? We've talked about the neuromuscular junction, which occurs right here, right, where the neuron meets the muscle. And we have a nice, happy story of events that occurs which allows for muscle contraction. Okay, a motor unit, though, is a nerve, okay, and all its associated muscle fibers. And a nerve may have branching points, right? So we have this purple nerve here, and it branches in a few different places. And so it can actually have a neuromuscular junction with quite a few muscle fibers. But it's still just one nerve. So one action potential from the brain that's strong enough should excite all of these individual muscle fibers. Wherever the nerve has a neuromuscular junction, it's going to cause contraction if a strong enough stimulus reaches that junction. And this is then our motor unit. So we have one motor unit in purple and then another motor unit over here. All right, and depending on the type of muscle, you may have a motor unit, maybe one neuron and one muscle fiber. Okay, but as you can see in skeletal muscle, it's generally a group of muscle fibers for each nerve. All right, so we're going to be talking about motor units with these different responses and how how a stimulus will affect that motor unit. So first off, we're going to talk about what happens with one motor unit. Okay, and what we call a muscle twitch. Okay, and this over time has a grade of tension, which is measured as a force in newtons. Okay, so we're measuring this as a force. Okay, and when a motor unit is stimulated, it essentially has this pattern. Okay, so it kind of actually has a less steep slope on the tail end than on the front end. All right, and then I'm actually going to grow, the first little part is going to be mostly flat here. All right, so we can break this into 3 sections with 2 lines. And if our stimulus occurs here at this point, okay, what's the stimulus in the motor unit? Okay, how is it perceived as the muscle? What's going on there? Right, so this is an action potential. This is an electrical signal sent as a voltage through a neuron to the neuromuscular junction. Okay? So what happens here at the beginning is nothing. All right, there's no observed force of contraction. This is called our latent period. But it's not actually nothing that's happening, right? What's occurring during this latent period?
The channels are opening up, all the calcium coming out.
All right. All right, so this is everything we talked about last time. All those steps of excitation takes a little bit of time for it to actually stimulate the muscle. So excitation is occurring at that latent period. But eventually, the calcium causes the tropomyosin to shift, and you now have interaction between the actin and the myosin, and that is contraction. So what's occurring at contraction? Yeah, right? So here we have our 4 steps of actin-myosin interaction. power stroke, detachment, cross bridge forming, and cocking of the head. Right. So you're going through lots of different interactions between different myosin heads. It's getting smaller. All right, and then lastly, we have relaxation. What's occurring at relaxation? Okay, first the stimulus stops. Yep, calcium reabsorption. I guess uptake would be the other term. All right, acetylcholine esterase. Is unbinding the acetylcholine to its receptor. And of course, the actin and myosin is no longer interacting. Esterase.
Oh, okay, there you go. Thank you, sorry.
Is that how you spell reabsorption?
What's that?
Is that how you spell reabsorption?
No, there's a B there. I knew something was wrong. It's hard to spell up here, guys.
It's hard to spell.
Acetylcholine esterase.
Yes.
ETYL.
Yep. All right. So that's what's occurring. That's a muscle twitch. That's one muscle twitch. Okay. Now we're going to switch scales a little bit. All right. And we're talking about one motor unit again. And we can have different forms of graded muscle response. Okay. So, if we stimulate the muscle like we just showed, we will have stimulation, right? And I'll get rid of yellow. Who put yellow back on there? Okay. So we've already shown if we have a muscle twitch, well, it's going to occur a little bit after the stimulation, right? And it's going to have some level of force in that area. But what we can do is, what if we start stimulating or changing the interval of the stimulation? So if we can control the stimulation, and we can put these stimulations closer together, where before it starts to relax, we stimulate it again. Right? And then let it relax. We can basically add more force by stimulating it before it can relax. So we call this wave summation. And we can do this okay, let's say we do this again, and we keep on doing it for more. At some point, however, we're going to reach a maximum, okay? And we're going to call this tetanus. Okay, so this is the point at which we can't increase the force. Okay, we can't increase the tension. So what's occurring here? How come we get an increase in tension if we stimulate it before it relaxes?
Can you zoom into that? That's really hard to read.
Which one?
Just in general, that's good. Thank you.
What's going on here? Okay, yeah.
Just like if you get the same amount of force from the stimuli every time, if you're starting at a higher starting point, each time you can have a fully relax your starting point, so a little bit higher each time.
Right. So before you are fully relaxed, right, or even barely relaxed, some of that interaction between actin and myosin is still in effect. Okay, so if we can stimulate it again, all those ones that were still interacting will be there, and then we can add more to it. Okay, so we're essentially recruiting more and more actin and myosin interactions if we can stimulate it close enough before it relaxes. So why does it level off? Yeah.
You only have so many muscle fibers to activate.
Right, so eventually you're going to run out of muscle. Okay, and it's going to then level off, and you're not going to be able to recruit anymore. Okay? All right, there's another funny thing that happens. If you stimulate, you give it a full, I'll put it over here. If you let it allow it to relax completely before stimulating it again, there is an observed increase. It doesn't occur every time, it maxes out, but it is above the first stimulation. Okay, does that make sense? We aren't increasing the stimulus. We aren't, we're allowing it to fully relax, so we're not getting this wave summation. But we still get a little bit of increase. And as we continue to do it, it doesn't go up anymore. So if we did it again, it would still be at that peak. All right, so this phenomenon is called TREP.
Yes, that would.
Okay, and what's thought here is maybe the muscle fiber is just warmed up a little bit. So if it goes from complete relaxation to stimulation, you'll get a response. But now that even though we let it fully relax, we still have the processes, I guess, right on the cusp and ready to be stimulated the next time around. And you get a little bit increase in it.
The data say that could be because calcium is pumped back into the subcutaneous reticulum by calcium pumps, and that takes a while. That you could have complete disconnection from actin and myosin, but still have calcium ready to start the process quick.
Right.
So is that like the priming or the warming up of the muscle?
Yeah, it could be that. And again, this is a hard sort of mechanism to study, but that's the thought. Calcium's right there ready to go the second time you stimulate it, and so it's more easily.
That's the warming up.
Yeah.
That's it being warmed up. Is there is calcium ready available?
Right, could be. But that is readily observed if experimentally this TREP phenomenon. Okay, so those are graded muscle responses. when we alter the frequency of stimulation. Let's see if I have, nope. Okay, and now if we look at a whole muscle, okay, so let's say we have a biceps muscle. Okay, there, it must be 11:30. There is a current there's a stimulation at which you can still get no response in the biceps, okay? And we can measure different stimulations in the voltage. So last time we were talking about the frequency, but if we can, let's say we start at 1 millivolts and go up in voltage. Okay, and when we stimulate this biceps muscle at 1 millivolt, we get no response. Okay, so we're going to do it again at 2 millivolts, and we still get no response.
Is it at the same point, like totally no response?
Yeah, 0.
Okay.
So I'm right at just above the 0 line, but I mean 0. Okay, and we do it at 3 millivolts, still no response. But then at 4 millivolts, we start to see contraction. So we would say then that at 4 millivolts, this is our threshold stimulus. The point at which we can get the biceps to contract.
It's not different for various muscles, is it?
It is.
Oh.
But you're jumping ahead of me. So we get to, and then what we'll see is a continual tension as we add more and more voltage. But then at some point, let's say at 8 millivolts, we get a leveling off. So at this point, we would call this our maximum stimulus. Anything below our threshold stimulus then is 1 to 3 or 0 to 3, right? This is our sub-threshold stimulus. Okay, but the biceps is a pretty big muscle. And so if we looked at a different muscle, let's say we looked at the brachioradialis. Okay, it's in that same sort of area, your arm. And we did the same thing with the brachioradialis, and we found at 2 millivolts, we got a response. And then, of course, as it continued, we got this continual response at more and more voltage. But if brachioradialis is a smaller muscle, what would you expect to happen? Yeah, I think what you guys said was the maximum stimulus is going to be much shorter. Yeah.
So does how high or low the maximum stimulus is have anything to do with like the amount of force that the muscle can exert? So,
Yeah,
like, you're
that's
stronger
exactly.
muscles.
Yeah.
So, but does that apply like, because you're saying this is the same for all of those muscles, so like, if I go to the gym and the bicep gets bigger, does the maximum stimulus stay the same?
Well, you could increase your muscle mass, and yeah, no, it wouldn't stay the same.
Okay, but it's
You're-
just more of the muscles.
Right. You'd have more fibers available for lifting those weights.
Hypothetically speaking, your quadriceps have a higher maximum stimulus than biceps, so they're generally stronger.
Right. Yeah. But it's better to look at these within an area, right? Essentially, you have a number of muscles that are working to move your arm. Some of them are going to do smaller movements, like the brachioradialis, and it's going to take less to stimulate them. Some of them are going to take more stimulus, meaning you're going to use them when you need them for something bigger, right? So if you remember that video we watched where the guy was opening the door, right? So that would be, the muscles needed to do that would be on that lower threshold. But if it was a steel door, you would need more muscles activated. So you would activate the brachioradialis and the biceps as you're trying to pull it. Yeah. So this allows you to have different muscles for different activities as needed, based on how big they are. Okay? But all of them, of course, at some point are going to be maxed out. You can't exert more force than the muscles that are available. Okay, so now we're going to switch. So those are our graded muscle responses. This is what happens with muscle fibers or the motor units as you stimulate them in different frequencies or at different voltages, which corresponds with essentially how much strength you can get out of your muscle. All right, but where is this energy coming from, which is what we're going to talk about next, okay? The first source of energy in your muscles, going back to what we know about the microanatomy, is where? Right, so you have ATP. We're going to write that on the right. I'm going to use a different. So there's already ATP on the myosin head, ready to be used in that cocked position. Okay, and if you just decide, I'm going to start sprinting right now, that's when you're at your fastest, right? It's not at the end of the marathon, it's like at the very beginning. Okay? And that lasts about 3 seconds. Okay, so it runs out pretty quick. So you can sprint for 3 seconds pretty fast, right? If we had a 3-second race, if we had a 10-meter race, okay, it would be hard to determine the winner because everyone kind of has that same sort of energy available. Okay, all right, for the next 10, 15 seconds or so, up to 10 seconds, right? Where's the energy coming from? Well, this is coming from something we haven't talked about, at least before, right? Creatine, phosphate. So what happens when you use this ATP on the myosin heads? All right, you get ADP and a phosphate, right? Yeah. So one of those phosphates breaks off and you use that energy. Well, creatine phosphate is another cellular enzyme that makes ATP readily available right there after that ATP is burned. So it has immediate source of ATP to replace this ATP and gives you another 10 seconds of ATP that's available. Okay? But eating creatine is not going to help you have more creatine phosphate. Yeah, but yeah, so this is kind of, the health world is filled with like misinformation. They take something like this. Oh, creatine phosphate is used for energy. All right, well, let's make a bunch of powders out of it and sell it to people, and so they think that they're going to get stronger if they eat it. But it doesn't work like that. You have plenty of it. It's there, creatine phosphate. You can't actually get more of it. It's not going to make you stronger if you have more of it. You need to make your muscles bigger, and this doesn't have to do with your muscles. Okay, so you can throw away all that creatine that you have in your
They advertise creatine is working differently though. They say like it pulls all the water from like everywhere else in your body and puts it into your muscles.
Baloney. Just kidding. I don't know. Maybe. But I would, I don't know, maybe that's true, maybe it's not. But I would like try and find the scientific paper where that happens. Is this based on science, or is this just some guy chopping in front of a YouTube video saying stuff, right? All right, so then for the next 30 seconds, you're kind of out of ATP that's immediately available, and now you have to make ATP. Okay, and the quickest way is through glycolysis. Okay, and glycolysis is where you take glucose and you break it into 2 pyruvate molecules. And from that process, you make 2 ATPs. All right, this is the simplified version. All right? But this does not require oxygen. Okay, so we call this the anaerobic. This is an anaerobic pathway. And you'll continue to utilize this in the aerobic pathway because you need pyruvate for the next steps in aerobic respiration. And pretty much everything after 30 seconds is all ooh, man, I'm doing good. Okay, I'm going to draw a line like this. Is all aerobic. Okay, so this is where we're going to take this pyruvate. We're going to run it through the Krebs cycle, which is then going to give us some substrate for the electron transport chain. Which is going to give us 32 ATPs. More or less, somewhere around there. They don't know exactly. Yeah. So, where is this glucose coming from? Well, it can come from blood. It can come from muscle. And then you'll also have stored glucose. How do you store glucose? All right, so you've got glycogen in your blood. And glycogen in your muscle. What if you run out of glycogen? Fat, right? So you can take fat and insert it, break it down and insert it into the Krebs cycle
Not like the
as well.
keto diet where you like get rid of all your sugars, you start burning fat for energy.
Yeah, so the keto diet, the idea there is if you don't eat carbs, then you're going to skip that step. You're going to go straight into oxidation of fats.
Would you say it works?
Yes.
Keto diet?
It works, but you also, if you follow the diet strictly, you're also reducing your calories. So if you're doing it to lose weight, it's probably because you're reducing your calories, not necessarily going into ketosis.
Like 2 weeks or less, 5 weeks as you started.
Yeah, most people that are doing it, I read it, I don't know. Yeah, there's a lot to talk about there. I'm not going to talk about it. Yes, you can lose weight on the keto diet. Is it good for you? I don't think so. Now, proteins. You don't see proteins on here. You can use proteins in the Krebs cycle as well. But generally, if you're doing a long-term exercise, your proteins are pretty much tied up in your muscles, right? In the things that are needed for your protein. And so you're not going to burn through proteins until you are like beyond exhausted, right? So you're mostly just going to do fat and glycogen stores, and your protein is all tied up doing the work of the contraction. So, how much is in each area? Like, do you start with fat? Do you start with glycogen? That is something that is still under study, and I've got an article here, and this is something that you could reference in your discussion. Substrate utilization during exercise in active people. Okay, so they looked at where the energy was coming from as people exercised, and they started with people that were just walking. Okay, so what I want you guys to do is just read the abstract, okay, and take 5 minutes to do that. Okay, and then we'll talk about, all right, well, where's the energy coming from on these different exercises? Okay, so go ahead and read it with your neighbor. Talk about, all right, where's the energy coming from? And then we'll discuss it together. You get 3 seconds.